Hello everyone, I'm Nilufar and today I'm going to talk about our clinical grade Treg manufacturing with a focus on GMP compliant cell sorting and also our Max GMP platform. As you might know, Miltenyi Biotec is at the forefront of pioneering clinical research with emphasis on immune therapy, tissue regeneration, and graft engineering. Our validated products for cell manufacturing, such as CliniMACS Prodigy platform and MACSQuant Tyto Cell Sorter, enable GMP-compliant cell processing and have been used in more than 50,000 cell therapy procedures. Let's have a look at our agenda for today. First of all, I would like to give you an introduction about regulatory T cells, then talking about clinical application of Tregs. I have three main topics for today: graft-versus-host disease, GVHD, rejection of solid organ transplants, and also autoimmune diseases. Afterward, I will go through our clinical Tregs manufacturing, talking about workflow 1, workflow 2, Treg culturing and activation, and also at the end, Treg analysis. And I would like to sum up with the most important points and closing remarks. Tregs are important subset of adaptive immune cells and control immune reactions for maintaining homeostasis in our body. And they have three main functions: suppression of overwhelming immune responses, which keep the balance and homeostasis in the body. The next one is suppression of immune responses against self-antigens and non-self antigens, which protect against autoimmunity and mediate the tolerance. And the third one is suppression of anti-cancer immune responses, which is not the favorite functionality of Tregs because it supports the tumor growth and needed to be considered during the cancer treatment. As you might know, there is no unique surface marker for Tregs, and they are characterized by the combination of surface markers. In general, Treg cells express CD4 and CD25 and identified by the absence of markers CD127. And the naive Tregs can be identified by the additional marker, which is CD45RA. Moreover, FOXP3 is an intracellular marker of Treg and an expression of this marker used as a proof of functionality of the Tregs. And Tregs classified according to developmental origin into three subsets: naive Tregs, peripheral Treg, and in vitro Treg. Treg has crucial roles in different types of diseases and provide novel therapeutic strategies for treatment of different immune disorders such as inflammatory diseases, tumor immunity, transplant tolerance, and autoimmune disease. This picture here shows the healthy situation that there is an immune balance between regulatory T cells and T effector cells. However, in some circumstances, this balance is disturbed, like GVHD, autoimmune disease, or organ transplantation, rejection, and so on. And the reason which underlies the immune imbalance are diverse. And adaptor immune therapy with Treg cells lead to the reestablishment of the immune balance. And it's the reason that why Tregs are very attractive candidates for cellular therapy. In the recent years, Treg therapy is one of the approaches for not only treatment of prevention and treatment of GVHD, but also autoimmune diseases and solid organ transplantation, which introduce some sort of new needs and criteria in field of clinical grade Treg manufacturing. But we have enough solution for all that I'm going to mention them during the presentation. Many of you are already in the field of clinical Tregs and are touching all the challenges in this field every day. And as you can see here, I've listed the main challenges, which are, for example, high chance of contamination because of open systems, inconvenient isolation procedure, maintenance of Treg after expansion, the functionality and phenotypic characterization is really difficult. Isolation of Treg subsets is very challenging, and also reaching the ultra pure Treg population is a big issue in the field. But at Miltenyi Biotec, we are trying to solve these challenges and support you with very high technology products, devices, reagents in GMP grade. And in this presentation, I'm going to present you most of our solution that we have in this area for you. And let's start with clinical Treg manufacturing workflow. There is a three-step workflow for producing clinical grade Treg, which consists of Treg isolation, Treg expansion, and also Treg analysis. And as you can see here in this slide, I've sorted all the needed products for this workflow that Miltenyi Biotec provide for you. Here you can see the two main workflows that we offer you according to your application and needs. But the main point that makes these two workflows different is related to the isolation step. As many of you might know, Treg is a rare subpopulation of T cells, around 1 to 4% of whole PBM cells, and isolation of them is really challenging. So recovery and purity of isolated Tregs are really critical factors that we will discuss it during the presentation. And workflow 1 and workflow 2 answer your needs. So let's start to go through each of them, but I would start with Treg isolation step and compare the method in workflow 1 and 2. In workflow 1, isolation based on CliniMACS Plus, which is a dominant and very well-known device in the field regarding to Treg isolation, and you can find it in most of the clinical Treg publications. CliniMACS Plus enable you to first deplete CD8 and CD19 cells and afterward enrich your CD25 positive cells. As you can see here, it is mainly used for production of cells in the field of GvHD prevention and treatment and final yield at an intermediate to high purity, approximately up to 96%, and the whole isolation process takes about 7 hours. As I have explained in the previous slide, CliniMACS Plus enables you to first deplete CD8 and CD19 cells and afterward enrich your CD25. It can be followed by sorting. That I would like to talk about it more after workflow 2. In the flow cytometer plots, you can see that by getting the high CD25 expressing cells, we can target Tregs, and after enrichments, we reach the purity around 92%, which is a great number for Treg isolation. Okay, let's summarize it here that using CliniMACS Plus CD25 system offers you convenient isolation process, high final yield cell number. After the isolation, you can see that phenotypic and functional characterization of Tregs are maintained. And with this method, we target polyclonal Tregs population. Very good. But two questions pops up here. That first one is how to isolate different Treg subset or antigen-specific Tregs, and how to reach the ultra-pure Tregs. They are the questions that I'm going to answer in the following slides. But first of all, let's have a look at the main challenges in the recent year in the field of clinical Treg manufacturing. The first one is naive Tregs production. Naive Tregs is a small cell population, and a high number of naive Tregs is needed for the new application. Ultra-pure Tregs. It's really stressful and time-consuming fellow sorting methods, which is not the favorite part of this workflow for Tregs because they are really fragile cells. Low capacity of fellow sorters are not really answering the need of the market as well. And GMP flow sorting, it's really hard to get all the approval for the clinical studies with the current solution in the market. Altogether, to solve these challenges, we offer you the workflow 2, which contain two main devices, CliniMACS Prodigy and MACSQuant Tyto Cell Sorter. These two devices help you to first to isolate Treg subpopulation and antigen-specific Tregs, and also enable you to produce ultra-pure Tregs. Using CliniMACS Prodigy LP25 process enables you firstly enrich your CD25 positive cells, secondly labeling of your target cells with fluorescence antibodies, and thirdly elute them in the special Tyto Cell Sorter buffer. And altogether your final yield is ready to be run in the cell sorter afterwards. And in this way you can isolate your ultra pure antigen specific Tregs. As you can see here in the chart, the main application of this workflow is related to the cases that scientists or physicians need antigen specific ultra pure Tregs, and they can reach more than 90% purity, which is a promising numbers. The whole workflow takes about 10 hours. Let's start with CliniMACS Prodigy. CliniMACS Prodigy offers a flexible platform for cell processing applications, enabling the magnetic separation of different cell types as well as customized cell processing protocols. As you can see here, Prodigy provides you a sterile, closed, portable, flexible, reproducible, with high safety and traceability system. And the specific process for Treg enrichment in Prodigy is LP25 process. In the following slides, I'm going to explain more about the here you can see the most important factors that you need to know to start the LP25 process in Prodigy. The starting material is leukapheresis, and you can see the total cells volume. Regarding to final products, you need to know that how many cells will be your final products, and also the elution volume and how you can elute it in the buffer to go to sorting your cells afterwards. And it's important to know that the process time is about 5 hours with 1 hour hands-on and 4 hours automated process. And on right side of this slide, you can see the reagents that can be connected to Prodigy. The process can automatically use all these reagents and run the process, and at the end you will have your enriched Tregs and ready to be transported to the sorter. Let's start to have a look at the whole process step by step. The first step is CD25 enrichment. Before enrichment, just 3% of the whole blood cells are Tregs, and after enrichment we reach to 30% of the whole cells. And you can see here very well that naive Tregs are very small fraction of the whole blood cells, and ultra pure isolation of them can be very challenging. Next step is fluorescence labeling of enriched cells. We provide you max GMP quality antibodies, and don't forget that all these steps are automated and no hands-on is needed. The last part of LP25 process is the elution of target cells in Tyto buffer, and then transferring the cells to MACSQuant Tyto Cell Sorter. Here you can see the labeled eluted cells with our GMP antibodies. There are some important points that, first of all, we provide you max GMP fluorescence antibodies for Treg sorting. We offer you services to design your own panel. We support you to optimize your cell sorting, but don't forget that our suggested panels that you can see here with respective GMP antibodies are already optimized in Tyto Cell Sorter, and it's just ready to use for you. Now I would like to hand over to Felix to explain more about MACSQuant Tyto Cell Sorter.
Yeah, thank you very much, Nilüfer, for that very interesting presentation so far, and also thank you for already introducing the MACSQuant Tyto. We're talking here about a multi-parameter flow sorter, which is equipped with 3 lasers, 8 fluorescent channels, and 4 scatter channels, and therefore provides quite some flexibility in terms of panel design. It's very suitable for clinical applications because the sorting happens exclusively within a sterile, closed cartridge. And this also means that there's no carryover or cleaning between samples necessary, as the whole fluidic system is always exchanged when you use a new cartridge. The whole thing works as an easy plug-and-play system, and also no droplets or detectable aerosols are produced within the cartridges. So it's very safe for the operators as well. Another benefit is that the sample can never be lost, as the cells never leave the cartridge during the sorting procedure. So let's have a closer look at the cartridge. Here you can see that it consists of 3 different chambers. An input chamber, where the sample of interest goes into, a positive collection chamber, where the positively sorted cells will end up, and a negative collection chamber, where all the rest flows through. A mixing propeller in the input chamber prevents the cells from settling down. The analysis as well as the sorting happens at a small microchip at the bottom of the cartridge. So I would also like to look into that in more detail. Here you can see from bottom view that we really are talking about a small MEMS chip. Here you can see that comparison. And if we look into more detail, we already can see it's sitting in the middle of the base of the cartridge and is connected to the chambers via different channels, which you can see here. If we zoom in again, we see that there is a sorting channel. The cells coming from the left-hand side pass the lasers, and in case a positive target cell has been identified, a magnetic pulse will open a microvalve, which then very gently and simply redirects the cell into the upper channel, and all the rest, as said before, flows through into the negative collection chamber. Let's have a look on how this really looks like. What you here can see is a strobe capture of the moving microvalve, which can open and close up to 30,000 times per second. As said, once the cells pass the 3 lasers and a positive target cell has been identified, a magnetic pulse opens the valve, which then very gently redirects the cell into the positive collection chamber. If we look at the whole concept of sorting, you can see that the air, which is coming from the MACSQuant Tyto instrument itself, is entering the cartridge through a sterile filter with 0.1 micrometer pore size. The second of those filters is sitting right before the input chamber. So sterile filtered air is entering the input chamber and building up a very mild air pressure, which brings the cell suspension through the microchip and then again facilitates the analysis and the sorting process on the microchip. So the positive target cells will end up in the input chamber, and all the negative cells, as well as the majority of the buffer, will flow through into the negative collection chamber. So why is the MACSQuant Tyto Cell Sorter such a good fit for clinical cell sorting applications? Sorting happens exclusively in the fully closed and sterile cartridge. There are no fluidics inside the instrument itself. The cells never leave the sterile sorting environment. Furthermore, the Tyto is currently approved for 10 different clinical trials, either in phase 1 or phase 2, and over 200 patient samples have been sorted to date. Last but not least, all consumables which are needed for sorting process on the MACSQuant Tyto are available in Max GMP grade and come with an extensive documentation for the discussions with regulatory authorities. And with that, I'd like to hand over back to Nilüfer, and she will discuss some more application examples, also including the MACSQuant Tyto and the sorting of regulatory T cells.
Thank you so much, Felix, for the very nice and comprehensive explanation about this great technology. And let's continue. And first of all, have a look at the efficacy of sorting of Tregs using Tyto Cell Sorter. For sorting of Treg and having ultra pure final yield, I would suggest you to use the 2-step sorting strategy. First, run the cells for debulk sorting, and then follow that with purity sorting. After debulk sorting, you can reach up to 83% purity, and after purity sorting, your final yield will reach up to 98% purity. Let's have a look at the whole process with some numbers. If the process starts with 1/4 of a locopack, which contains 2.5 billion cells, after Prodigy LP25 pre-enrichment process, we can reach approximately about 38% enrichment, which lead to the total Treg numbers around 7.7 E7. And after cell sorting using Tyto Cell Sorter, you can reach about 98% Tregs, and the important part is the number of naive Tregs, that you can reach 2.6 million naive Tregs. And it's good to know this workflow is already used by some of our customers, scientists, physicians in the field, and they are going to start a clinical trial very soon. After cell isolation, we need to expand the Tregs. As many of you know, expansion of Tregs is very challenging. However, we provide you with proper mediums, cytokines, activation reagents, and so on to facilitate these steps. Let's have a look together. Expansion of Treg is mainly done in a manual and open system. However, Miltenyi Biotec is offering you a special service which is related to the customized automated system. As I have mentioned before, CliniMACS Prodigy offers a flexible platform for cell processing applications. So our CAP team can design a process for your Treg expansion, or even a process which covers the whole workflow, and make the workflow easier for you and complete the whole workflow in a closed system. So to get more information, please contact us. For expansion of Tregs, proper medium is one of the key factors. TexMax GMP medium is specialized for optimal cultivation of human T cells and Treg cells. It contains pharmaceutical-grade human serum albumin, optimized formulation with or without phenol red, and QC functionality test is doing on every batch, and filled in flexible bags, which makes it easier to connect to CliniMACS Prodigy as well, and they offer the customized format under requests. Three other key components are Max GMP Expect Treg Kit to expand Treg cells after a CliniMACS CD25 enrichment and maintaining FOXP3 expression. The second one is Max GMP rapamycin to inhibit the expansion of conventional T cells and also to enhance the proportion of expanded Tregs FOXP3 positive cells in culture. And at the same time, one of the very, very important factors is the cytokines. Max GMP IL-2 is our suggestion for you, that you can see its specific activity in comparison to the other commercial products. And again, for getting more information about all the details, please visit our website or feel free to contact us. Here you can see the post-sorted fraction of Tregs with 98% CD25 positive and CD127 negative, and also 92% CD25 and FOXP3 positive. And after cell expansion for 2 weeks using TexMax GMP medium, Max GMP Expect Treg Kit, rapamycin, Max GMP IL-2, we reached to 98.3% CD25 FOXP3 positive cells, which is a great yield according to the expansion rate. Purity and functionality of your final Tregs are really great. And please don't forget the point that culturing of Tregs is really challenging, and it's really critical. It's donor dependent, and it's really dependent to the fact that if you're expanding your healthy donor samples or patient samples, and it really depends on your manual protocol. There is no standard fixed protocol duration. It really depends to your plan, to your application, and it varies between 14 to sometimes 36 days. And yes, they are the factor that you need to really take into the account and set up your own experiments. The last but not the least step is the analysis of Tregs during the cell manufacturing process and also at the end of the process to control the quality of your final yield. You can analyze your Tregs phenotypically or functionally. As you can see here, there are a sort of markers, and according to your experiments and Treg cell subset, you can design a panel and characterize your cells. Don't forget that we support you to design the best panel according to your needs. At Miltenyi Biotec, we offer you the big family of MACSQuant analyzers, which are fully automated flow cytometers with different properties. As you can see here, I just wrote down very in brief, but please get more information from your local salespeople or just visiting our web page. For phenotypic analysis, we have two solutions for you. The first one is the Treg detection kits, which are optimized antibody kits with pre-titrated antibodies that developed as a single one reagent solutions. The second solution is reaffinity recombinant antibodies and biodi antibodies conjugation, which are highly pure and has lot-to-lot consistency. We offer you panels of antibody for phenotyping your Tregs with surface and intracellular markers. And here I put two examples. For example, in this 8-color panel, we detected Treg surface markers. And in the other example, I just showed you the staining of the intracellular markers, which are really challenging, but we have good antibodies and also buffers to help you to stain your intracellular markers and analyze your Tregs with the surface marker and also intracellular markers. For the functional analysis of your Treg, I would recommend you our Treg suppression inspector kit. In this kit, we have Treg suppression inspector, which is a polyclonal stimulus, MACSQuant, coupled to alpha CD3, CD28, CD2. And these MACSQuants are optimized to stimulate responder T cells to proliferate. However, when the T cells are co-cultured with functional Tregs, their proliferation can be suppressed. Here you can see better that when your two responder cells receive the stimulus, they start to proliferate. But when the two responder cells are co-cultured with your functional Tregs, their proliferation stops and suppressed with Tregs, which is a proof of functionality of your Treg. I'm almost at the end of the presentation and would like to just remind you again that we cover the whole workflow and we have solution for each steps. If there are any questions about really like detailed of products, devices and so on, please feel free to contact us and we can discuss together and present you good solutions according to your application and your needs. And also you can use the information that is offered to you on the web page regarding to the Treg clinical scale, research scales. We have good brochures, application notes, references list, scientific reports, and also regarding to the each products and devices, you can get enough information on our web page. And with disclaimer, I would like to end up my talk and thank you so much for your attention.